Well, thank you very much for that kind introduction. And it's an absolute pleasure to be here today, and I really appreciate the opportunity to speak with all of you. I was asked to talk about the human microbiome research at NIH. But before going into that, I'd actually like to give a little bit of background on the human microbiome and throw in a little bit of nutrition and why I believe this is so important for the nutritional community to be thinking about the microbiome. As was mentioned, I'm from the Office of Dietary Supplements, but what I'm going to say are my thoughts and opinions and not necessarily those of ODS. And also you can see my email address if anybody has any questions after this presentation or would like a copy of the slides, please feel free to email me. Also, we were asked to include a disclosure slide. I have nothing to disclose, except I'll be talking about nutrition research at NIH, and I work at NIH. I should say microbiome research. But one thing that I would say is I really have a strong belief that understanding the interrelationship between diet and microbiome will really start to provide some of the tools for understanding why some people respond to changes in diet and others don't. And I passionately believe that this is a very important emerging area of science. So a little bit about background on the human microbiome. We are actually considered to be superorganisms. We are a composite of species. In addition to our mammalian or eukaryotic cells, we also have bacterial cells, archaeal cells, and in fact, there are 10 times more microbial cells in the body than human cells. These microbes live in different niches. The majority of them live in our gastrointestinal tract, and we talk about the microbes in a different niche as a microbiota. For example, the gut microbiota is 100 trillion organisms that live in our gastrointestinal tract. The microbiome is their collective genome, and in fact, there are 100 times as many microbial genes as there are human genes. And in fact, we're considered to have a metagenome, which is a combination of both the microbial genes and our mammalian genes. These microbes do serve many different important functions. They provide the ability to harvest nutrients. They produce additional energy otherwise inaccessible to the host. A good example is the fermentation of soluble fiber to short-chain fatty acids, which are an important energy source for the colonocytes. They produce vitamins such as biotin, folate, and vitamin K. They metabolize xenobiotics. A very good example are the heterocyclic amines that are formed in food during cooking. And in fact, they can metabolize these to inactive metabolites. And they prevent colonization by pathogens, and they assist in the development of a mature immune system. This slide shows the development of the human intestinal microbiota over time. It was initially believed that when children were born that they did not have an initial microbiota until birth, but we're now being increasingly realized that they're actually exposed in utero to specific microbes in the amniotic fluid. But the additional colonization really does depend on the mode of delivery, and it's different between those infants that undergo vaginal versus C-section delivery. In early childhood, you start having more of a stabilization, and in adults, it's when it's the most diverse and very stable, and then you start losing diversity in the elderly. And that's actually shown on this bottom slide. In the infants, you actually have the highest variability and the lowest diversity. In children, by about age 3, it's very similar to the adult microbiota. It changes during pregnancy, and you have new types of bacterial species emerging, such as proteobacteria and actinobacter. But after pregnancy, it emerges back to the general adult. And you start losing diversity in the elderly, and you start seeing increased numbers of Bacteroidetes. There is evidence that diet can actually influence the composition of the microbiota, and it shifts the microbial community. In fact, in this study, they took 17 obese patients and put them on a number of different diets, including one very similar to the Atkins-type diet, where it was very high in protein and low in carbohydrates. And what they found is when they were on this Atkins-type diet, there was actually a shift in the microbial community and the specific metabolites. This diet also led to a decreased proportion of butyrate in the fecal short-chain fatty acids, reduce the Roseburia or Eubacterium rectale group of bacteria, and reduce the concentrations of fiber-derived phenolic acids. And these are actually very important metabolites in terms of gut health. So even though you might lead to weight loss and stuff, you could actually have detrimental effects that are happening to your colon. One of the questions is how quickly can you actually have shifts in the microbiome in relation to diet? And in fact, in this study, they looked at effects of diet on microbial composition within days. They looked at 1, 2, 3, and 4 days after a diet that was either exclusively plant-based or exclusively animal-based. And what you can see is that short-term consumption of these diets composed entirely of animal or plant products actually altered microbial community structure and overwhelmed inter-individual differences in gene expression. And this is actually, I guess you can't point, but it's actually shown if you look at the hierarchical clustering, you can see all the red, which is the animal-based diets clustered together, whereas all the green, which is the plant-based diets clustered together. So they're clustering by the diet within a day rather than by the individual. It also modified specific metabolic pathways, which are shown in the bottom right. But there's actually a two-sided relationship. We talk about the fact that diet can influence the microbiota, but it's also important to look at the other side of the equation, and that the microbiota can actually influence the response to dietary components. In fact, bacteria can generate new metabolites from dietary components, and many of these bacterial-derived microbial metabolites are actually what are exerting the biological effects. Some of the foods that are metabolized by gut bacteria are shown on this slide, as well as their bacterial metabolites. And many of these metabolites are actually more biologically active than the parent compound. And also, for many large polyphenols, it's actually the microbial metabolites that are exerting all of their biological effects. There is evidence that you can have shifts in the microbiota between in different diseases and physiological states, such as obesity. In this study, they took 12 unrelated obese subjects and compared them to 5 lean individuals, and they found is with obesity, there was a significant increase in the amount of firmicutes and 90% less Bacteroidetes than the lean individuals. Moreover, if they actually put them on a weight loss diet, either a carbohydrate-restricted diet or a fat-restricted diet, monitored them for 1 year so that they lost up to 25% of their body weight, they actually saw that there were increases in the amount of Bacteroidetes that correlated with changes in weight. And in fact, there is evidence to suggest that you have microbial dysbiosis in a number of diseases, and that you can have interactions between diet and the microbiome to influence disease risk. For example, dietary carnitine, which is in meat, and dietary phosphatidylcholine and choline in eggs are actually metabolized by microbes in the gastrointestinal tract to form trimethylamine oxide. And this metabolite is activated in the liver to form trimethylamine oxide, and there's a significant correlation between the amount of trimethylamine oxide and the risk of a number of diseases, including heart attack, stroke, and death. And in fact, animal studies have shown that the molecular mechanisms for this are through increased cholesterol transport. So I hope that I've actually convinced you, as a bit of background, that there's a very important relationship that both the microbes and dietary components and energy interact and impact both the numbers and types of bacteria, as well as specific dietary components and metabolites can influence disease risk. And it's with the belief that there's a relationship between the microbiome and disease that led to the initial Human Microbiome Project at NIH, with the idea of saying, is there a core microbiome? What is a normal microbiome, and how does that vary during different disease states? So here's some information on the Human Microbiome Project. The initial Human Microbiome Project was from 2008 to 2012, and I have the website listed if you'd like additional information. And the first phase really was a survey of the microbiome. The question was, who's there? It was a $175 million project funded by the Common Fund and a number of NCI offices and center. And the question it wanted to ask is there a core microbiome that's the same among different individuals? And how does this vary among different body sites? And by understanding this, then maybe can we understand what goes awry during disease? So it really had two main projects. The first one was a healthy cohort study where they used 300 females and male patients that were 18 to 40 years of age, and they sampled sites from 5 major sites, including the nasal, gastrointestinal, oral, and urogenital tracts. And they collected 15 samples in men and 18 samples in women, and they gave up to 3 different samples in 2 years to look at changes over time. And subjects were not allowed to be taking any antibiotics, probiotics. The other part was what was considered the demonstration projects. And these were 10 different studies that tried to compare the microbiome at different sites between healthy individuals and normal individuals with the idea to say, are there differences in the microbiome between healthy subjects and different diseases so that we can figure out what might be going wrong? And what they found was that in healthy American adults, the microbial community composition in each part of the body is unique. And in fact, when you look at the different sites, you see that they cluster together, that the gastrointestinal tract clusters separate from the oral and from the urogenital. And for each person, it was estimated that there were about 1,000 different bacterial species and 2 million bacterial genes. And in fact, when they looked over the 300 subjects, they identified over 10,000 species and 8 million genes. But even more interesting is there was no single microbial species that was present in every individual. And although 2 different individuals may have similar abilities to metabolize carbohydrates, it would be different bacteria that's having this effect. And in fact, there is no core microbiome. That's actually shown a little bit better on this slide that looks at the microbial composition of each body site, and this is looking at the phylum level. Each vertical line is a different individual, and as you can see, there's a lot of variability among individuals, as well as variability among different body sites. In contrast, when they looked at the genetic potential of each microbiome, that's looking at their metagenome and the potential biological pathways that are influenced, you can see that there's significantly less diversity. And in fact, there's much less diversity both among individuals and among body sites, suggesting that it may be this genetic potential which is more important. And just to summarize that a core microbial composition may not occur with the host phenotype. However, a core metagenomic and/or functional profile may occur with host phenotype. So one of the questions is how does the metagenome relate to transcription of microbial genes? And actually, how do you develop the tools to start being able to look at this in epidemiologic studies, to start understanding the relationship between the metagenome and the metatranscriptome? And this pilot study looked at both the metatranscriptome and the metagenome using samples from the health physicians study. They took 8 individuals, and I'm sorry, I can't read the slides at all, so I'm trying to remember what's on the slides, but it's impossible to see these. They collected samples both from saliva and from the stool, and they sampled them in 3 different ways. They either, for the saliva samples, they just had them frozen, whereas for the stool samples, they either had them frozen, they put them in ethanol, or they put them in RNA later. And they collected DNA from the saliva samples and both DNA and RNA from the stool samples to be able to analyze the metagenome of the saliva, the metagenome of the stool, and the transcriptome of the stool. And here's some of the data from that study. When they actually looked at the microbiota composition and looked at the 10 most abundant microbial taxa and the metagenomes, they found that it was highly variable if you look at the vertical lines. In contrast, when they looked at the 10 most abundant microbial metabolic genes in the metagenomics of the subjects in the cohort, it was not variable. And the last panel shows the 10 most abundant expressed microbial genes in the metatranscriptome of subjects in the cohort. And it was somewhat variable. It was significantly more variable than the metagenome, but significantly less variable than the microbiota taxa, suggesting that individuals actually do regulate their gene expression in the microbiome. And we need to understand how that occurs. So as I said, the human microbiome initially gave a lot of information about the distribution and diversity of different human flora-associated bacteria, but it really didn't give that much information about the biology of the microbiome, how the microbiome interacts with the host, and how the host actually responds to the microbiome. And while the metagenome is the genetic blueprint of the microbiome, what is the most important thing to be studying? What functional properties do we need to look at to know how it's being regulated? Should we be looking at transcripts, proteins, or metabolites? And this really was the focus of the second phase of the Human Microbiome Project. The second phase, which is known as the Integrative HMP or IHMP2, is really focusing on what are the microbes doing. And it's a $25 million project, again funded by the Common Fund and a number of NCI institutes and offices. And really it seeks to look at the biological properties of both the microbiome and the host to understand microbial composition and utilize multi-omics technologies such as transcripts, proteins, and metabolites from both the microbiome and the host. It's utilizing longitudinal cohort studies and integrative data sets as a community resource. And pretty much it's funding 3 different studies, and I'll go into these in a little more detail. The first is looking at the dynamics of pregnancy and preterm birth. The second is looking at the dynamics of inflammatory bowel disease, and the third is looking at the dynamics of type 2 diabetes onset. So this slide shows the study looking at the dynamics of pregnancy and preterm birth, and it's utilizing 2,000 pregnant women to better understand the relationship between the vaginal microbiome and other microbiomes and how these relate to birth outcomes, as well as to the development of the microbiota in the infant. They are collecting a lot of additional information, such as diet histories. They're collecting a number of different samples and utilizing a number of different assays to look at multi-omics in both the host and from the microbes. The second study is looking at the dynamics of onset of inflammatory bowel disease. And pretty much it's hoping that if you can get an understanding of how the microbes interact in the gastrointestinal tract, then maybe you can better understand what's happening during flares, what is the molecular changes that's happening, and how to utilize this information both potentially for prevention and therapy. It's utilizing 3 different cohorts, both pediatrics and adults and healthy controls. Again, it's getting a lot of background information, including diet histories. And one of the interesting things in this study is they're actually doing biopsies to be able to look at the gene expression in the host rather than just the microbial populations. And the third study is looking at the dynamics of type 2 diabetes onset. And pretty much it's utilizing 60 subjects that are at risk for diabetes and collecting samples every 2 to 3 months when they're healthy. But if they're exposed to a respiratory virus or some other environmental stress, they're collecting samples more frequently to really understand how does respiratory viruses and other stresses influence the microbiome, and does this change glucose tolerance or insulin resistance, and really understand how the microbiome changes as you start progressing from prediabetes to diabetes. Information about the integrative human microbiome can be found on the website, and the link for the website is shown here. You can find information about when data will be released, because this is meant to be a community resource, and it's meant for the whole scientific community to have access to the data sets. Information about the current members of the different projects can be found there. as well as information about the DAC, or Data Access Center, and where it's being stored, as well as information about if you want more details about the 3 different projects I just spoke about. Well, I was mentioning what NIH is doing in terms of funding research, but one of the other important things that NIH really tries to do is bring people within NIH, different program directors, together to really say, where is the science now, where is it going, and try and get the different institutes and centers to work together. And one of the ways it's doing that is through the Trans-NIH Microbiome Working Group, or TMWG. It was established in 2012, and it's extramural program staff from 16 institutes and centers. And its mission is meant to be a forum for microbiome-related investments at NIH, to identify gaps, needs, challenges, and opportunities, to share upcoming funding opportunity announcements, to coordinate joint funding of applications, to discuss development of joint FOAs, to discuss development of microbiome review panel at CSR. And in fact, we did just find out about a week ago that the Center for Scientific Review will have a standalone study section to evaluate microbiome grants, which was really a big thing that we've been pushing for a while. And it really serves as a central source for the external community. Information about this working group can be found at this website. And as I said, I'm throwing a lot of websites on here, but if anybody wants information on these, feel free to email me. I'll get you the information so you don't have to worry about writing it down. But it really also has the key points of contact for each of the institutes and centers, and current microbiome-related funding opportunity announcements for people that are interested in doing research in this area. There are a number of other federal working groups. One is the Probiotic Working Group, which is more trans-agency. In addition to people from NIH, it has members from USDA and FDA, and its goal is to advance the base of probiotics and prebiotics. And we have developed 2 funding opportunity announcements related to prebiotics and probiotics that are currently in the NIH guide, and hopefully will be released within the next month. There's also a Microbiome Obesity Working Group, which is a subset of the Obesity Research Task Force, and it initiates and facilitates activities for advancing our knowledge of the contribution of the human gut microbiota to obesity. And the last group is the Microbiome Quality Control Project. And this is actually composed of both intramural NIH investigators and HMP investigators. And it's really a collaborative effort to comprehensively evaluate methods for measuring the human microbiome. So many different laboratories are doing things in different ways. What is the best way to collect samples? What is the best way to process them? What is the best way to bioinformatically look at the data? It's trying to look at all the different methods and try and come up with what are the best ways to do things in the future so that we can start comparing results between different laboratories and start having the tools to be able to do this type of research better in epidemiologic studies. You know, I've been talking a lot about microbiome research, and that's what I was asked to focus on, but there are many other resources out there for the microbiome community. The one is the AGA Centers for Gut Microbiome Research and Education. And this really is meant to be a virtual home for all the microbiome research by the American Gastrointestinal Society. A number of universities, also that from HMP investigators, also have developed microbiome programs. There are a number of journals out there that are focused on microbiome-related research and are starting to publish stuff on this. But I think of what might be the most interest to this group are a number of citizen science groups that are out there. Probably the most famous right now is the American Gut. And this is 32 investigators that said, we really need to understand what is the relationship between microbiome and health, what is the ideal microbiome, how is it influenced by environmental factors, particularly diet. And what they're doing is they're taking 10,000 individuals from the United States, including cats and dogs, and they really want to get an understanding of what is the ideal microbiome. Do we really know? How can we influence this? How is it related to disease? And every one of you out there, if you want to be involved in this, if you want to know what your microbiome is, you can send them a sample along with $99. That's how they're funding it, through crowd science, and they'll let you know about your microbiota. Another example is Explore Your Microbiome, which is a very similar type of initiative, but that's throughout the world rather than just limited to the United States. So what are the take-home messages? We need to realize that there's a two-sided relationship between diet and the microbiome, and we really need to take a metagenomic view of our dinner plate. Rather than just our traditional view, we need to think about what are the microbial metabolites, what are the different food components that are metabolized by the microbiota, what are those metabolites, and what is their relationship. We also need to understand both the microbial community and what the microbes are doing. Where in the past we really focused on what microbes are there by using sequencing, and moved to what is the genetic potential by using metagenomics. We really need to better understand how they are regulated and utilize metatranscriptomics, metaproteomics, and metabolomics to really understand their functional ability. And finally, we need to have a healthy dose of skepticism. There's a lot of things out there saying this probiotic does that, this probiotic does something different, it's great. Well, you really need to ask yourself 5 questions. Can the experiments detect a difference that matters? Does the study show causation or just correlation? What is the mechanism? How much do experiments reflect reality? And could anything else explain the results? So I'll finish with this slide. I want you to remember that bacteria are your friends. I have my email address again if you have any questions, and I'll take any questions if there's time.
We have time for one question. Please.
So Connie Weaver, Purdue. Thanks, Cindy, for that overview. I think the most likely promising research that you described is when you go from pre-disease following the changes through disease to see what happens. Given that you say there's no core microbiome, it seems like to learn anything about diet, we have to take that similar philosophy and take it through interventions and see what changes are made, because this business of going 2 groups, high fat and no fat and different populations, I don't think it's going to lead us very far.
I think the important thing for your background and what you said on that is that there's no core microbiome, but there's a core metagenome. And so the question isn't what microbes are changing, is what metabolic capacity of those microbes are changing over time when you go from disease, go from health to disease, and then how those are modified by diet.
And through diet.
Yep. Well, great. Thank you very much.